Bioprocessing technology is nowadays a key enabling technology for the new bioeconomy. In that context, we know since many years how to ferment microorganisms or cultivate cells. However, the recovery and purification of delicate bioproducts becomes, and has been for a long time, a current challenge. Modern industrial biotechnology make use of microorganisms for the production of a variety of different bioproducts. These bioproducts, they can be generated in large amounts in stirred tanks or bioreactors where a controlled environment is actually provided. The scale of that bioreactors can be moved up in order to create large amounts of such compounds. Once biomass has been generated in bioreactors, there is a need to separate the solids from the liquids. This can be done utilizing different techniques, one of which it is called French press separation. This is a filtration technique that essentially utilizes membrane cassettes and filtration aids to produce a supernatant or a liquid free of any suspended cells, that liquid being used for further processing. Another common technique to exert separation of cells from the surrounding media is to use centrifugation. During centrifugation, cells are exposed to forces that make them to decant or sediment, creating again the possibility for us to obtain a completely clarified liquid that can be subjected to further processing, as we are going to see later on. Once that products they have been isolated in a soluble manner in a liquid phase, we need sometimes either to concentrate them or to fractionate the crude mixture in order to gain a level of purity and concentration. This can be done utilizing, for example, tangential flow filtration, utilizing hollow fibers in this case, or sometimes flat sheet cartridges as well.
So this machine, ÄKTA avant FPLC, is to further purify the already fractionated samples into separate components.
Once that our product has been properly purified, we need to stabilize such a preparation. This can be done, for example, generating a powder out of the concentrated product, utilizing spray drying, or further stabilizing the liquid by rotary evaporation. The pharmaceutical industry has since long shifted partially from standard chemistry into biologicals or biotechnological methods. That has made possible for them to generate products like monoclonal antibodies, which are used in the treatment of cancer and other very difficult to treat diseases, but also the generation of new vaccine forms, as we have seen in the recent pandemic times. New products like plasmidic DNA or messenger RNA are gaining more and more acceptance in the market. In the case of the food industry, we have seen a convergence between pharmaceuticals and food technology. The food industry is looking more and more for specific additives, bioactive compounds, and so on, in order to improve the quality and the properties, the nutritional properties, the biological properties of the new food that they are delivering to customers nowadays. Standard chromatography works on the basis of utilizing a packed bed of beads. This technology has been around for many years, several decades, but nowadays we see a shift into a different type of materials. For example, fiber type of materials, fiber types of absorbents. These absorbents, they present many advantages, like for example, higher flow rates, higher productivities, and the ability to deal with a repertoire of products which goes beyond the traditional protein manufacturing that we have seen. The future of bioseparation technology and downstream processing lies nowadays into a full integration with the bioprocessing train, the whole bioprocessing train. On top of that, we are adding a digital layer to it, so digital twinning, for example, in order to create more robust, sustainable, and profitable bioprocessing scenarios for the modern industry.